So before we get going, let's give the speakers another round of applause for a really great morning session. Okay, is this on? Are we live here? Good. We'll just pass it back and forth if you're okay with that. So I'm going to take moderator's privilege and ask the first question. Try not to absorb too much time. And then I definitely want to hear questions from Twitter and in the audience and wherever they come from. So I was thinking as you were talking, there's sort of a unifying principle kind of across all the talks. And you can certainly not answer any of the questions. You can always beg off to. But you know, what the unifying kind of concept to me is that you're bringing information that's not traditionally brought to patients, through either genomics or, through SNP testing or through the patient portal, and you're kind of breaking the mold. Obviously, the traditional patient goes, like Gena was saying, patient goes to the doctor, and they're the font of all information. And I think Jeff would acknowledge, and other people, everybody on the panel, that's creating churn in our regulatory world, because there's always this third party that's kind of sitting over and above us, like either payers or regulatory people saying, we need to approve that or not approve that. So I guess, first of all, just kind of get your reaction to, do you find that to be a major pain point? And if so, sort of what your thoughts are about a path forward through sort of working with these third parties, shall we say, whether it's governmental agencies or payers that are sort of rapping your knuckles a bit, saying don't give that information back to people, or I mean, or tell me I'm all wet if I'm misperceiving something. So
it's really, really important that we understand the impact that we're having on people by giving them this information. And there's a variety of different viewpoints, and we can polarize a room in a second, I think, by bringing them up. But it's good to have these discussions, and I think with respect to the process we're going through, the FDA has the obligation to protect Americans and to make sure that the information that they're getting through devices such as ours are not putting them in harm's way. And of course this is a take my word for it kind of a statement, but of course we have that same principle that's guiding us. And so, we put a lot of time and effort into developing our product to take people through an educational process and to allow for easy, quick access to information, but to let customers dive deeper and get to the source materials to understand the science behind the information we're presenting them. And education is a big part of what we believe in to help people best interpret the information that we're presenting them. It's definitely a brave new world, and we're going to see this in more and more areas. I mean, just mobile health in general and digital health is just an area that's under a lot of scrutiny right now, and we're definitely disrupting what's happening. And I hope that it's for the better. We were founded under the notion that it's best for individuals to own their own information, because that's empowering. And if you have to go through someone else to get that, it's a big barrier for a lot of people, as some of the data represents that I showed earlier. Some people just don't want to get it in a regulated environment because they don't know what those people are going to do with that information. So it's a great question, and there's a lot of answers that are still coming, I think, related to that.
It sounds like education is one of the key, trying to wrap a good educational package around your institution.
So with us, like 23andMe, we believe that patients should have access to their own health information, and we believe in the transparency of providing that information to patients. And we're facilitating that and really doing that through the healthcare professional. And because of healthcare reform, doctors need to do this. So for us, our biggest barrier, overwhelmingly, patients want access to this information. They are logging in, and we have an 800 number where patients call us for technical issues. And overwhelmingly, we hear from patients, I just went to the doctor yesterday and my labs are here. So they're really, really excited about this. And the surveys we're doing, we're finding that they overwhelmingly want this information. Our biggest issue and our biggest challenge, the federal government has said that this is important, and they're requiring doctors to do that. Our biggest issue is just the change management within the healthcare process, and for healthcare professionals to to get comfortable with actually giving patients access to this information. They're really worried about the number of calls they're going to receive and what's this going to do. We have a lot of issues with the rollout plan and actually getting providers to actually turn on the service. And once they do, they're shocked because patients don't call. They're just excited to receive this information. It hasn't increased the workload, but there's just this perception that it will. And so that's our biggest barrier really to implementing our service, and it's just the change management of getting healthcare professionals comfortable that this is happening.
I think I agree with what's been said, and the third parties, regulators and payers, I think largely have everyone's best interests in mind. They're trying to help people be safe and all of those things, which is obviously critical and important. I think patients want to be active participants in this. We're at the forefront of, I think, a revolution in the way that medicine is practiced, which is largely because of the Human Genome Project. And as genomics begins to play a larger role in every aspect of medicine, patients are going to become more and more actively involved in that process, and they should. I think that some of the barriers I think the major barrier is exactly what Gena and Jeff said. It's education. It's education not only on the part, we need to educate the payers. They don't understand why a certain drug would be given in a certain situation. We need to educate the regulators. I think there's a lot to be done there. And we need to educate the providers. There was recently a poll in our major professional journal of medical oncologists called the Journal of Clinical Oncology, where they asked medical oncologists across the country are they comfortable in dealing with genomic data for their cancer patients, and 80% said they are not comfortable in dealing with genomic data for their cancer patients. And so that's eye-opening, but I think it's true, and so there needs to be education on the patient level, on the provider level, on all levels, and as we get through that, I think access will begin to open up.
Sorry, thank you. No. So with our few minutes left, are there any questions out in the audience? So,
I'm just curious about the economic benefit of treatments that you prescribe, those targeted treatments. We had such an amazing response on the imaging. Trying at least to figure out what was the economic benefit to the insurance companies also that don't have to take a lot of other chemotherapy.
so I'm going to repeat the question for the folks online. The question was to Lincoln, I think, around, in addition to the sort of the patient outcomes and improvements in care, what are the economic benefits?
Thanks. Yeah, that's a great question. We have spent a lot of time looking at that. We're going to publish our data in a formal format soon, but I'll just give you some sneak peeks into the broad principles that we've identified. I've been surprised to see that drug companies are really smart. So the costs of these targeted therapies, even oral drugs, are surprisingly very similar to the cost of infusing an IV drug, if you look at it on a monthly basis. And I think that shouldn't be surprising if we consider how these companies, are incentivized to figure out their price structure, right? They want to, of course it's going to be similar to what the, other standard currently is. But where the impact is really dramatic in providing targeted therapies is in, ancillary costs. So we don't have patients spending time in an infusion chair, requiring an infusion nurse to tend to them for 46 hours during an afternoon. And then we don't have nearly the side effect problem. So we don't have patients getting admitted to the hospital with neutropenic fever and other complications or dehydration or whatever other problem they have from some of our cytotoxic therapies. So the economic impact is big and it's dramatic, but it's not at the level of the cost of the drug. It's at the level of all of the, ancillary factors. Good question.
Yes, up next.
The question I had is really a follow-up to the first question that you posed, and that is, how do you deal with the fact that you have Hipaa regulations? Most of the targeted therapeutics that you're using are not approved You have to have special FDA. I mean, all of this takes time. How do you deal with this in the molecular tumor board discussions, and how does that interface with your institutional review board and with the FDA? It seems like that's a very complex series of events that has to occur for the patient to receive the drug.
And a follow-up to Lincoln is, how did you set this infrastructure up and really perform this hat trick to get patients drugs that are off-label, that are clinical trials? I mean, how did you actually do that? Who paid for it? What's that infrastructure you alluded to?
And then we'll let Gena and Jeff talk about Hipaa a little bit too, hopefully. So.
Well, it's a good question, and it's not easy. It hasn't been easy. And basically, it consists of having close and extensive conversations with the payers. And so in most cases, we've been able to get the payers to provide the drug for patients when these are patients that don't have other options and they have failed, approved therapeutics. Using a drug off-label in that setting has been not an issue. Then we have a whole class of patients where we've only been able to get them access to drug through clinical trials. So we have a whole array of trials, including, Tim and I have talked about the Novartis signature trial that we have available that has a whole bunch of targeted therapeutics that are available based on a patient's genomic alterations. And so we've been able to get a lot that way. So it's been a combination of lots of close conversation. Payers want to have these conversations, and so do regulatory agencies. And that's how we've been, it's just been a coordinated dance, so to speak. Yeah. It takes a lot of FTEs, I can tell you that.
I don't think I have anything significant to add other than just talking a little bit about Hipaa. And so we are a Hipaa compliant application. We're a business associate with the clinic partners that use our service. But how we've managed Hipaa, so we're allowed through business operations, and because clinics need to give patients access to their health information, we just do that. For patient education and for other services that we provide to patients in a very personalized way, we have patients opt into that and sign a Hipaa authorization. So we're really about providing the right personalized information to every patient, but based on their own personal preferences. And so we ask what they want and deliver based on their needs and wishes.
Just a quick comment on that. As a company, we're direct to consumer, so we're not dealing with sharing information amongst stakeholders in the healthcare environment yet. But to the extent that we do our research, and to the extent that privacy is a concern there, we do all of the research in a de-identified fashion, so there's no concern of patient disclosing who a certain patient is. And just a quick comment about the economics of personalized treatment like this. I mean, I think that's kind of like half of the whole hope and promise of personalized medicine, is that we can try to hopefully cut the fat, if you will, and get more quickly to the appropriate treatments that patients should be on, whether that's identifying risk early in their life or late in the game, but saving them the trouble of having to go through a bunch of ineffective treatments. That's really half of the hope and promise of doing all this.
Can you shout? Yeah. And I'll repeat it. Okay, so again, for the folks on the interwebs, there were a couple of questions. One was around what are the implications of knowing a lot more additional genomic information in terms of sort of reproduction and having future children and such, and maybe selecting in a strong way against certain mutations and such. Is that affecting sort of reproduction decisions? And there was a follow-up question on patients knowing, maybe being, I'm putting words in your mouth, but over-empowered so that they're gaming the system with regard to insurance, or stacking the deck against insurance companies, I guess. So.
Okay, well, two fabulous questions. Thanks for asking them. I don't have the perfect answer for probably either of them, but I can have some thoughts. The first, with respect to your case of testing, early in life or even before life to see what genetic risks may may be involved. That's a super fascinating case. And I think, again, way to polarize a room, there's many different ways we can debate this from an ethics perspective and a medical perspective. I think it's not too far of a jump from what may already be happening within the context of genetic counseling today. I mean, it's a little different because you're very distinctly pinpointing a specific condition that could be at high risk. But with genetic counseling as it occurs today and family planning, carrier status screening is not uncommon, certainly within certain populations of people. And people are making informed, to the extent they can, informed decisions about their reproductive choices based on things that they know to be that they're carriers of. So cystic fibrosis being a perfect example. So if you know that your risk of passing cystic fibrosis onto any offspring is, 1 out of 4 or something, that's going to impact the way you decide to move forward. And I think it's similar, although yours is much more dramatic, a case of being informed about your reproductive choices. And I won't come down on either side of that now. I think it's an interesting thing to watch as we move forward. The second question related to insurance, are they gaming the system? Well, maybe. I mean, that's an interesting way of putting it, but we all, to a certain extent, do that based on our family histories, right? So, we're looking at germline risk for disease, but, it's not too far from just looking at your family history and saying, well, there were some pretty amazing cases that were presented from Intermountain about, generations of folks with pancreatic cancer or hepatobiliary cancers. And, those folks would probably, be acting out of their best interest to, think about screening for those conditions and, just using that information in a helpful way to guide their health moving forward. And whether that's taking out certain insurance policies or getting screenings or doing things like that, then they're just being informed and making smart decisions, and I think we'd all want to be empowered in that way. To the extent that insurance companies want this information, they absolutely want this information. They absolutely want this information. And that's a further testament to the power that this information holds, I think. But as I mentioned before, as Americans, we're protected in certain ways against this information being used against us. And hopefully there'll be more and more legislation, guiding this. But they absolutely want it, and that's one of the reasons we feel very convicted that you should own the information and that we shouldn't have to provide it through a healthcare provider or through an insurance company. And it should be your decision as to whether or not that information gets shared with anybody. Now, the FDA might have something else to say about that, but that's our position.
So I'm sorry to repeat. If I understood the question right, you're pointing out the fact that we have all this new genetic information. Some of it's related to single gene mutations, but the majority, 75% or more, is non-single gene diseases. So how, was your question more kind of mechanistically, like how are we mining this data? Is that kind of a... Okay, well, maybe I'll pass it down to Lincoln and Jeff.
Well, in the case of cancer genomics, you're absolutely right that there are modifying factors that play a role, and that's one of the things that we constantly discuss in the molecular tumor board. We see that, gene A, B, or C represents a good target. What about gene, the other 3 or 4 genes that we identified? What role do they play, if any? Are they drivers? Are they passengers? What do they represent? And I think we're so early in this that we're trying to figure that out. I will say that that's been one of the major benefits of having a live molecular tumor board, is that we can parse those things out somewhat. I also think it's a real-time learning experience where we're already learning what to do in one situation based on what we thought we were doing 6 months ago that is now different, or what we learned from those cases. We don't always pick the right driver to go after the first time. That's happened a few times where there are 2 potential driver or actionable mutations you use a targeted treatment for the first one and you see progression, and then you use the second one and you see dramatic responses. So it's hard to know. I think the bigger question you're asking, though, is what about those inherited, genetic modifiers, and what role do those play? I think we're so early in the game from a cancer genomics standpoint that we're not ready yet to start considering the bigger picture and the more distant modifiers. You're right that there's lots of data and possibilities of doing that. The family histories are extensive, and we can start to do that, and I think that's part of the plan down the road. It's still a little early, and I'll let Jeff and others comment.
My only quick comment is we are really early in this process of getting, from a technology perspective, is starting to collect the data. ASCO recently came out with guidelines about the importance of capturing family history, cancer family history in a certified EMR. Navigating Cancer created that 4 years ago, and so we are capturing cancer family history and meeting those guidelines today, which gives us the opportunity to start to collect that data and then to combine that with other data sources so that we can really start to understand. But we are really in the beginning stages of this.
Yeah, there's so much data being collected now and in such an efficient way that, eventually sort of merging efforts will be one way to just catapult us even further. So, if we had the genetics piece and Gena had the health records piece, and putting that together could be really powerful, right? I mean, we have over, in some cases over 1 million different SNPs on each individual tested, and almost 700,000 individuals tested, plus 750 million phenotypic pieces of information based on self-report. And just to give you an idea of what we do at our company, thankfully we've graduated past the fax machine. We have computational biologists who are constantly combing through our data and constantly running GWAS to just discover what's out there. And the nice sort of notion of having this hypothesis-free opportunity to just discover is, is that we can rapidly, find things and discover things all the time. And so in some cases it's too much information to sort of start parsing out what's, what are our priorities? Like, what are we trying to tackle first? And so in some cases where we have developed distinct cohorts, like with our Parkinson's cohort, we're approached by a funder, and sometimes that's a pharmaceutical company or just someone with an invested interest in discovering more about what's causing this disease or how patients may respond to certain medications who have this disease. Then we can actually put together a well-developed cohort of people and ask specific targeted questions that are put together by survey methodologists and scientific advisory panels, and really drill deep into one disease area. But we're constantly mining our own data, and will, forever, I assume, as long as we're gathering it.
So the question was, in this era of collecting this data online and through, patient portals and through such, without a professional data collection agency, if you will, whether that's a CRO or whatever, how useful or how confident, how do you deal with the fact that probably some of that data is not absolutely correct and such? I'll just say one thing as a moderator's comment about big data. So I read a really interesting article in the Wall Street Journal about 6 months ago about what is big data, because as I mentioned at the beginning, I was curious myself because I didn't realize I was working on little data my whole life. But anyway, big data, apparently what distinguishes it from little data is 3 concepts according to these authors. One was the so-called N equals all phenomenon. So you're not, as opposed to statistics, you're not just sampling you're just getting all the data. So you're looking at the whole genome. You're not looking at one spot on a chromosome. That's the N equal all. The other is to Tim's point about the concept of sacrificing perfect data and perfect accuracy for the fact that you're getting it all. So you're realizing you're getting a fuzzy picture, but it's still probably a correct picture, hopefully. And the third concept was a little bit more abstract, but it was around the idea that you're just finding associations, but not necessarily understanding why things, the data's not telling you why something's happening, which is what we're going to hear about later today, I think more about the why. But this is just, there is a correlation. So I don't know if you want to comment to Tim's question about data accuracy.
So my comment on data accuracy is, first of all, before we even talk about patient-reported data, I'll just make a couple comments about just data accuracy within the EMR. So part of high tech is about getting data into structured formats and requiring doctors to have data on various things in structured formats. So EMR data, over the last decade, doctors have been adopting electronic health records, but what they've been doing is putting that data into notes fields, and so none of it has been structured data. And so we haven't had great data liquidity, just as we deal with the clinical data within EMRs, and that is starting to change because of high tech. Stage 1 had requirements around structured data fields. Stage 2 takes that to a higher level of degree. It is requiring vendors to use the same nomenclature for the codes. So, for labs, we will all use LOINC codes, for example, and so that will allow connectivity and data liquidity between systems so that we actually have better data from a clinical perspective. So lots of work that is underway to make data liquidity possible, to have systems talk to each other, and for them to talk to each other using the same nomenclature, and also have that data in structured data fields. So improvements are happening there as we speak. And then from a patient-reported perspective is, there are a lot of things that patients know about themselves that we ask them today that we take as the right data. So if you ask a patient, and you're just writing that down, or you're putting that in the EMR. So I think that there is the piece of patients can report on certain components, and of course that will need to be validated and confirmed with a healthcare professional, but I think that does have a place in healthcare, and I think we'll learn more as we move forward, but there will be that validation component, and we'll start to understand what components of what patients are reporting that we can say is the standard data set and what needs to be validated as we move forward.
Yeah, that was well put, and a good question too, Tim. I think, well, for one, just having, practiced medicine, there's a lot that goes on in your encounter that's you're basing on self-reported information from the patient, and it makes up a great, proportion of what ends up being in the chart, right? The patient's not going to be able to report back to you what their diagnosis code was, but you're gathering a lot of information from them that ends up being translated into the medical record. So I think we trust the patient a lot anyway in these encounters, and that's represented in the medical record eventually. The other thing is that when we're doing these big data mashups and trying to look at how the genome is matching up with the phenome, we've done some trials using EMR data, and until you get to a certain point in time, it's a mess. It's a literal mess. It's very difficult it's totally unstructured prior to, I don't know, maybe the last couple of years, and it's getting better since then. But until there's a lot of standardization that comes down the pipe and everyone's using the same format, it's going to be a big mess. And so patient-reported data is maybe the best thing we have going. The other thing is that I know that this was prior to my joining 23andMe, but I think it was a couple years ago we did publish a paper on patient self-report and the accuracy of those reports. I think it was with our Parkinson's group, where we actually did look at medical records and compared that to what patients were reporting for a certain subset of our questions, and there was a lot of, it correlated very, very highly. I can get you that paper if you're interested.
I think we have a Twitter question.
So one of the people who's been watching the live stream has sent in a question saying, can you please discuss the quality of the specimens used, what tissues, whether they were initial occurrence or reoccurrence, and how those differences may affect the response to personal treatment?
I think that's a linking question. So.
Yeah.
Did you get that? Or
Yeah. The question is about specimens and what types were used, and we've used all types. We used, fresh tissue from a fresh biopsy. We've used archival material in the form of what's ubiquitously known as FFPE, which stands for formalin fixed paraffin embedded. And this is the format that almost all biopsy or tumor specimens, or really any pathological specimen, is preserved over time in an FFPE format. And we have not seen a significant difference between tissue formats in terms of the alterations that we identify or response to treatments. We haven't seen any differences that we can detect. And I think sequencing technologies are sufficiently powerful now that we can tolerate more and more tissue formats. But the biggest ones, fresh and FFPE, are totally usable.
Any other online questions? Third row there. So I think the question was generally, what type of panels do you use for the genomics testing, and what do you do about Medicare coverage?
Yeah, you've asked a hard question. We use the same panel. It's over 100 genes, and it tests the gene end to end, as opposed to just hotspots. It's also capable of detecting copy number variations and deletions and some chromosomal translocations. So I think it's much more powerful than the typical hotspot mutation testing that we see popping up here and there. Particularly for the copy number variants. That we're finding that represents, copy number aberration is about 1/3 of all actionable mutations that we're finding. So to be blind to that is a significant problem. All of the various payers that are not Medicare, the reimbursement has been very good. Medicare is not reimbursing, but has said that they, I think that you're going to see people, including Foundation Medicine, some of the other big ones out there, are going to start billing Medicare towards the end of this year. So that's kind of the current state of the art. I think that's evolving very rapidly.
Any more questions yet, Tim? So again, for the online folks, the question was around, to Jeff, around the sort of the compare and contrast BRCA1 in this 23andMe panel versus the myriad and other commercial tests.
Another great question. First, just to clarify, because I had a bunch of people ask me, just even during the break, well, you guys aren't still in business and selling kits and things like that. Just to clarify, we still do sell the personal genome service. The personal genome service today just consists of a complete ancestry composition, which is intriguing for a lot of people. And also you get your raw data. So you get all those, you get the SNP readout for I think our current chip has something along the lines of 500,000 to 750,000 SNPs reported. So you can get access to all that. So you can still get your BRCA result, so to speak, in the raw data form. We just can't interpret your risk for you related to that. How do we differ from companies that are actually sequencing an entire gene? We differ in a lot of ways. One is that we have a very efficient way to just look at single point mutations, so SNPs or single nucleotide polymorphisms. And with the BRCA in particular, there are 3 alleles that are the 3 most common amongst the Ashkenazi Jewish population that are related to increased risk for breast cancer. We test for those 3 most common alleles. Now, what other companies, the value of sequencing And what other companies such as Myriad and others are now doing is they're looking at the entire genome, and they can find a lot of what we would consider to be low incidence variants. So if you, if you picture a chart with incidence, there's the first three alleles that have the highest impact on risk for breast cancer, and then there's a distinct drop-off and a very long tail of probably hundreds of different alleles that can impact your risk for breast cancer, but the incidence of those is incredibly small. So, hopefully that's getting at an answer to your question, which is using BRCA testing through 23andMe is probably a good first pass for a very efficient and inexpensive look at your risk for breast cancer. Having a negative result from 23andMe does not mean that you don't have an allele that puts you at risk for breast cancer, but a positive result is positive, right? Because you do have one of those three most common. So in the case of the study that Dr. Franke did with the BRCA results and looking at utilization, post-result and impact on anxiety and such, those patients or those customers of ours that ended up having risk-reducing surgical procedures had confirmatory testing done through another service where they sequenced the gene. So I think that I don't want to say anything is standard of care because this is all still pretty much on the leading edge, but it wouldn't be uncommon for a physician to look at a 23andMe result and then want to back that up with a sequencing of the gene. It's just that in today's world costs thousands of dollars and may not be justified on the front end. So that's sort of the context in which you can think about BRCA.
Thanks for that. I'll make a quick little editorial comment too, just from being in the pharma industry and working through this whole genetics information revolution. And that when I was involved with collecting genetic samples from patients, we just, what's the right word, perseverated and anguished over what they're, and there was all this theoretical concern, and I'm guilty of it. I mean, this is why we have to be careful about sort of making sort of blanket decisions or judgments about data, because I was guilty of thinking that too, like, well, if we collect data, we'll lose half the patients on our clinical trials because they'll be scared of being on a trial with genetic data. And it turns out, if you just ask people, they're like, no problem. I want to be on the trial. It's a cool idea. And they don't have any problem with genetic data. So I was completely wrong in terms of what my expectations of how scared or sensitive people thought genetic data was. And so, again, there'll always be a spectrum of opinions and concerns, obviously, but my suppositions were wrong. And then when I actually did 23andMe, just in full disclosure, and I'm not paid to say this is just my personal opinion, but I felt I got the sense of empowerment, and that I had my data, and it did make me just look at my health more closely. I really didn't have didn't have BRCA1 mutation possibility, for example, of course, but it was very, again, it wasn't scary to me. It was very empowering, as you say. So, yeah.
I didn't want to put this as part of my presentation because it may come across as being kind of marketing-ish, but I do want to let you guys know that we do have an educational program for providers through 23andMe, which allows healthcare providers, which is a very loose term in my mind for eligibility purposes, to get their own testing through 23andMe in a complementary fashion. So you can talk to me afterwards, or you can visit our website and look at if you go to the provider program page, so 23andme.com/provider program, as part of our educational initiatives, because it is a very educational process to go through it yourself. And there's actually been at least 2 papers published on this by other institutions, looking at the ability for physicians and healthcare providers to understand the interpretations of this data, having gone through the testing process themselves versus not going through the testing process themselves. And there's a pretty dramatic jump in understanding if you've gone through it yourself. And so we feel obligated to use that as a resource for healthcare providers, and I'd be happy to send healthcare providers a free kit so they can go through the process themselves.
I guess just to tag onto the final tag there, that I think what we're learning too, as we look at this panel and talk about these subjects, is it's really kind of a tapestry. So that's the other point I guess we're learning from. And I would say the same thing, having done the 23andMe test, it does what it does, but it's not the only test out there. And obviously Gena's site is a whole other complementary set of information and knowledge. It's kind of a knowledge base, really, in a way. And then you go to Lincoln and you're going to get very specific information about your tumor, and they really don't highly overlap that much. So we're learning that this kind of data era is really, like I said, a tapestry of different types of testing and genomic data and such. So I think I saw one question. Yeah, I'm sorry, second row here. So the question was, is there any type of data that we're talking about amongst the panelists that you have made an affirmative decision not to share? Maybe we just go down the line, and I don't know if Lincoln has a...
In the cancer setting, not really. I mean, we share, occasionally we come across alleles or specific alterations where we don't really know what their clinical meaning is, and so I don't really focus or dwell on those, but I don't try to, withhold any information. I think this, particularly in the cancer setting, we share everything we find.
So we are providing a service on behalf of healthcare professionals, and so we give healthcare professionals the ability to customize what they're going to share and what information they're going to provide to patients. We have tried to, our view is that patients should have access to their own information, and so we have tried to we've tried to help healthcare professionals know that that's a good thing for patients, but ultimately we let them have full control of what they want to withhold or what they want to release to patients. But the other thing that I'll say about that is Obama just signed into law a couple of months ago the ability for patients to get access to their lab information without their physician. So now patients do have the ability to go get all of their lab information not through their physician. So there is a huge push to patients getting access to their information, the transparency of that information, and a big push to provide more information to patients than less.
Another great question. So we obviously look at, a lot of different SNPs that could be related to a lot of different things. We have a certain threshold. Back when we were doing health reports with interpretations of disease risk, we had a pretty high threshold for what we would consider to be established findings based on the published literature. So we wouldn't report on things that fell below that standard as far as, looking at what other scientists and other research had discovered about associations between specific, SNPs or alleles and disease risk. Beyond that, though, we have made a conscious decision not to report, for example, or not to test for things like Huntington's. That's just obviously there's a lot that goes into finding those results out and what implications that could have on an individual. So that's one case I can think of where we've elected not to do that. There are also some recent efforts that are likely to be published regarding and there have been various position papers published for sure about the reporting back of incidental findings, particularly in the context of genetics. And a lot of this has come out of the genetic counseling world, and they have very strong feelings about what should be reported back and what shouldn't be reported back and how it should be reported back, with respect to incidental findings. So there's a body of literature about that if you're interested in looking.
Okay, so if I understood it, the question was around maybe interactions between maybe non-genetic factors. I think you mentioned the brain and these genetic markers that we're talking about here. So I don't know, you want to talk about that, Mr. Cancer Biology?
Yeah, it's a good question. I think unquestionably there are some environmental factors that affect how genes are expressed. lots of hormonal factors, local and distant forces can affect how genes are expressed or not expressed. Specifically, whether the CNS, the brain, can affect how genes in other organs are expressed. I'm not as familiar with data on that, or that there is significant data in that regard. But there are certainly, lots of hormonal factors and other things. As far as causing mutations and, heritable or at least transmittable alterations in the genome, I think that's not as clear right now. I think gene expression data is clearer in that regard.
Yeah, nothing really to add to that other than obviously, I don't know if this point came across well enough in my presentation, but genetics is just one small factor as far as your overall risk for disease and other things. So the environment plays a big role in that.
Yeah, no, I'll just echo in that, when I was training, we always had this triangle of genetics, environment, and there's another leg that nobody ever talks about. It's called stochastic or random, because sometimes things happen to you just because of dumb luck, to be elegant about it. But the great thing about getting some information, at least one leg of that triangle, is it makes you start to think about the other legs, like you're talking about the environmental, and I think you tried to stress that, Jeff, in your talk, but it makes you think about things that you should be thinking about, your diet and your cholesterol and things like that. So I think that's why it's not scary to have genetic information. So, yes.
Sorry to answer these questions, but I sent out a conference call. Two questions. One, Jeff mentioned earlier that genetic data
So around like pharmacogenomics and that kind of molecular signature for response. Sure. So again, to repeat the question, there was, I guess the first part was around not just healthcare insurance, but other types of insurance, mainly life insurance, effects of genetic, or I guess protection, if you will, of non-discrimination based on genetic data. And then the question also was, is the use of molecular signatures to predict sort of treatment response dead? I think I would say emphatically, no, it's not a dead science or not a closed science, so to speak. However, I think there was a period of, I can speak from experience growing up, that there was a period of time we thought there would be just tremendous numbers of these genetic markers that would just kind of fall off of the tree and then predict all these drug responses and such. I think in all candor, that's not turned out to be the case, that there's a handful of these that we've identified, and they're they're working their way, or they're already into labels, to drug labels, like the metabolizing enzymes, and obviously for things like the targeted drugs, those are, you could call those response markers. But I would say a fair characterization is there's only a handful of those. Actually, we discovered one when I was at Schering-Plough, which predicts a response to interferons and hepatitis C, which is the world's strongest one, and it predicts about 50% of the response, so it's not even all of the response, obviously. But it looks like right now it's kind of a lot of smaller effect markers that are affecting, kind of adding up to your general response, and they just tend to be harder to find because of statistical reasons. So I'll let Jeff see what he has to say. So.
I'll just comment, I'll intercept the microphone here, comment on expression data and molecular signatures. I think that we don't hear as much about it with the advent of next-gen sequencing and other genomic approaches, but it's still active and I think very applicable, and the best example of that is some of the commercial tools that are available to determine whether a breast cancer patient should or should not get adjuvant chemotherapy following surgery. And MammaPrint or Oncotype DX are examples of those testing services that use, gene expression data, RT-PCR and other technologies to determine, help guide management of patients. So I think that technology, and using signatures is not dead and is still relevant. And I think we'll start to see some overlay between, genomic data and gene expression data.
With respect to the pharmacogenomics, I agree with Mark. There's a handful that have kind of risen to the top and have actual FDA recommendations and label changes. But I think clinicians in general are still trying to figure out how best to use that information, how we can get that information in a timely fashion to actually impact prescribing patterns and therapeutic options. Some of the ones that make the short list that you may use in your practice are warfarin sensitivity is one that we're trying to figure out how, and that actually has an FDA label on it. But trying to figure out how best to use that information to impact care. And then Plavix is another one, your likelihood to respond to Plavix as a blood thinner. A couple of the other big ones, the interferon and hepatitis is a big one, as Mark mentioned. Also abacavir and HIV treatment. And the statins and the myopathy that can be experienced by certain individuals is also another one that's up there. With respect to insurance discrimination, at a federal level, as I mentioned, we're all protected for discrimination at the workplace and with health insurance. That leaves open some exposure potentially on disability insurance and life insurance. And my understanding of that is that they are managed at the state level as far as regulation in that regard. But obviously there are a lot of folks who are interested in getting that level of protection at the federal level as well.
Next year, we got to get another microphone, right? So, but anyway, it's been a great session. I'm sorry we're kind of at the end of the time and probably exhausting our panelists, but I really have enjoyed it. And I have a couple of housekeeping comments, and then we'll close. Lunch is right there through that door down the hall, and I'm getting the text message that it's ready. So I'm using my smartphone tech right now. And the other comment is that if you want to do a tour of the VCOM really impressive facilities, you want to just have a guided tour, you need to meet kind of underneath this, or next to the skeleton, we'll say here, and you'll get further instructions about how to do that tour. So with that said, I really appreciate the participants of Jeff and Gena and Lincoln, and I really appreciate your participation, and let's give everybody a round of applause for All right, we're dismissed. Thanks.